Thanks, Fred, and that was a great discussion yet again. So yeah, so really the rest of the time that we have here is really going to rely on all of you for discussion to kind of, based on everything that we've talked about, let's identify, where are the gaps, and these could be gaps in knowledge, these could be gaps in funding, the types of research that maybe you're able to get funding from NIH or other sources, but having difficult, or the type that you're not able to get funding from other sources that may be a good fit for the foundation or our partners. Other gaps like, thinking about regulatory challenges. I would like to ask Crit Moonen to talk about some challenges he's been having regarding pharma, as more clinical trials are getting going and there's a need to have access to the immuno-oncology agents in combination with focused ultrasound. There's some challenge there in terms of how to get those agents. And so I've asked, Crit had brought that to my attention. And so I wanted to give him the opportunity to talk a little bit about that. We can have a discussion about that. I'm sure some of you are also having challenges there. Maybe we can come to some solutions or potential solutions about how we as a community can help address some of those issues. So Crit, do you want to talk a little bit about that?
Yeah, sure. Thanks, Jessica, and thank to the foundation and CRI, I think, to provide this common forum. I think that's great. We are preparing for to provide you a little bit of the background. We're preparing for a clinical phase 1 trial on histotripsy together with checkpoint inhibition, and that's a follow-up of the study, the preclinical study that of Avinash Iranki and co-workers that I think that yesterday was discussed during the preclinical session by Tatiana, and especially by Tatiana. Now, in this study that we are planning, our oncologist Karin Suikerbuik has contacted a few of the pharma to see if they would support us with free checkpoint inhibition inhibitors for this phase 1 trial. And even though we have good contacts with pharma, this is we got rejected, and so this is what I wanted to share with you and see. This is, of course, the cost of these. If you buy them, is they run close to, let's say, $80,000 per patient. So these are very significant. So I just wanted to know if other people have similar experiences, and also whether there is anything that we could do maybe together with the foundation and CRI to see if we can. My hope is that once we have some good initial data, that then support for us and also for other groups would be more easy to obtain. But we don't know what the exact causes of this are, or the exact background is. I just want to share it with you and see what you think of it.
Does anyone have any? I'm sure some of you have had similar challenges, or maybe you've had successes. Dr. Bullock?
Yeah, Crit. So we had 2 trials with anti-PD-1. It's pembrolizumab in both cases from Merck. I know in the first trial, the breast cancer trial we're doing, breast 48, that was, we had to write an investigator-initiated trial to them, which was reviewed by them. But that was at a very early point when pembrolizumab came on board, and they were very enthusiastic about trying it in different modalities. AM003 also has pembrolizumab on it, but I can't remember if Dr. Slingloff and Dr. Dengel had to write a proposal to them to get drug or not. But that is commonly the avenue when I've spoken to the scientific liaison officers for various companies. They just, and BMS in particular, just says write a proposal to them, and they consider the drug at that point. That's the avenue we've generally pursued, and it's not always successful.
Yeah, that's also the one. Thanks, Tim. That's also the approach that we actually took. We wrote a proposal to them, and they considered it. There was quite a few discussions, but finally they decided not to support it.
Well, I think also with AM003, though, it's an improved, we're using the focused ultrasound in an improved setting for pembrolizumab. So it's particularly the melanoma patients now, when we've got breakthrough or prior to therapy. So that's one way of angling it. If it's standard of care in that sort of patient population, then you can potentially integrate the focused ultrasound. I would defer very much to a protocol development expert in this space before I say too much.
And I would say from the foundation perspective, we've been in it's been a goal of ours, particularly this year, with all of these clinical trials coming on board and with challenges that folks like Crit have identified us, to try to form partnerships with pharma in general to get them to better understand focused ultrasound and the potential. We've had a challenge. I mean, we've gotten a lot of first and second meetings, and it hasn't really gone on to anything substantive. So because our thought was that if they could see the larger potential, it's not just one individual site coming to them sort of ad hoc, can we get some drug, but the potential of this field to grow. So it doesn't mean that we've completely struck out, but it has been a challenge. I do wonder if Jill, or I know with your vast experience with CRI and collaborations with pharma, if you have any advice on kind of how to try to help establish some sort of a partnership for our community.
Yeah, I mean, this is not something unheard of. I mean, years ago when we started our what is now our clinical accelerator, was really due to the fact that investigators are having trouble getting the drugs they wanted from biopharma. And one of the reasons we created the model we have now was to work in partnership with the investigator, the not-for-profit, and the pharma or biopharma company. It takes a while. We've been lucky. When we present, I think timing is a lot to do with it. And when we started this, AstraZeneca became one of the first pharma companies that we worked with at CRI with our clinical strategy, because they were kind of behind BMS and Bristol Myers at the time. And it was that we were presenting them that here we were funding this trial. It was another shot on goal for them. It was a way for them to see, kind of a combination therapy of CTLA-4 and PD-1, and they provided us the drug for free, and we had but it was a major collaboration. It became a collaborative agreement with legs to it, in terms of that we were going to share the data with them as hoping to jumpstart. I think you have to, if it's not going to be approved as an investigator-initiated trial, which is the most likely way to do it if it's a single institutional trial, you have to develop a relationship and see that you're doing things to the standard that would be the pharmaceutical standard. I think a lot of pharma have kind of I'd say gun shy in saying that a lot of the individual investigator-initiated trials do not meet the pharma standard, and even if data comes out, they don't really trust it, and it means they have to repeat it if they really want to bring it into their development path. So I think having trials done at industry standard, so that the data, they can say, okay, this is kind of another shot on goal, and we believe this, and now we want to take it into our development path to, to really do more with. So as I said, timing's everything. Right now with the PD-1s, a lot of these are coming off patent in a few years, and I think there might be less interest to doing trying new combinations, and with them, you might try getting if you're looking for PD-1s from a lot of the other smaller companies that have come on behind the major pharmas that may have interest, that they see, okay, this may be a new pathway for them with focused ultrasound. It might be a way to use their drug in a new way that could open up a whole market for them. So I think that might be something you should try, but it's developing these relationships. They take time and energy, and there's also always lots of change at Pharma too. So you can have one relationship and you think you're going really well, and next thing you know, the entire team is gone and you're down back to starting at the square one again to develop a relationship on this thing. So it's that it's a tough go, but I think Jessica, you're right in terms of if you can somehow present it that focus ultrasound has this network of people with experts in focus ultrasound. There's a market for that. There's a potential to be used in combination with immunotherapies. Delivering all of that collectively might have more weight than just one-off investigators asking for it. So
Thanks. That's really great. Dr. Wayne, did you have something to add?
Thank you very much, Jessica. I'm very honored to and really grateful to be part of this discussion, and really also want to follow up on Gio's points, and the question you raised. For like investigator like us, we develop so-called platform technology. Now we can do, for example, prostate cancer as a target, but we would also like to extend this to other like a cancer or tumors as targets, for example, pancreatic cancer, breast cancer. I don't know whether they are finding, opportunity or support from, the foundations to help us, because obviously those different cancer types will be quite different domains for us, right? We don't have much experience. So that's one layer. Another layer is, for example, we already demonstrate for certain type of cancer, for example, prostate cancer, we can demonstrate, nicely in animals. And there is a gap from animal work to show the efficiency to the clinical trials or preclinical trials. So I don't know whether there are also, mechanisms from foundations who can help us to, fill this gap so that those technology demonstrating animal can be, extended to the clinical trial, preclinical trials.
Yeah, so just for your awareness and for those who are less familiar with the Focus Ultrasound Foundation funding, our sweet spot for funding is really in the translational preclinical space, as well as kind of first in human clinical trials. So, so the type of work that you're discussing, I think, fits in there nicely. So we would say that things that are very, very sort of the fundamental science is not necessarily our wheelhouse, although we do fund some things in that area, but the real basic science hasn't been the real interest of our donors, who are more interested in the kind of translational side of things. And so that has been I think that has been a gap within the focus ultrasound field, for where that research hasn't been as likely to be funded at some other places. So that's been a good model for us, as well as, again, the first in human clinical trials, I think, are really helping to de-risk these new ideas for industry for further commercialization. So in that case, we would fund trials at various institutions, UVA, University of Michigan, Stanford, and that would be essentially 10 patient trial, maybe, helping to get some of that early clinical safety and preliminary efficacy data that could then be used for larger scale trials that would likely be funded by industry.
That's beautiful. Thank you.
Thanks. Yeah.
So if there's, I think this pharma discussion, maybe we can just continue thinking about ways that we might be able to work together as a community, and ways that the foundation could help in that regard. And we're going to still try to form these partnerships. So, it seems like this is definitely an area that would be helpful for us to kind of continue down the road in. So what about other, other big gaps or big challenge areas that you all are facing, where you think that we as a community working together, again, perhaps with the foundation driving efforts, could be helpful? Are there any that you can think of? Tim?
So I'm wondering about the logistics of multi-center trials because of the issues of patient recruitment. It's still a somewhat exotic intervention, focused ultrasound, and I think we don't have the oncologists on here that could speak to this more directly, but I get a sense there's a lot of sort of infusion-based systemic therapies of first line for many of the patients we would like to recruit for our trials. And is the equipment, I'm showing my ignorance here, is the equipment difference at different institutions a barrier to doing multi-center trials? I mean, we've talked about having a central repository for material analysis, et cetera, which is great for harmonization. But to what extent can we have harmonization between different institutions at the equipment level?
I mean, that definitely can be a challenge. You know, one of the talks in our pre-recorded content from Gail Tahar was about the kind of treatment reporting guidelines. That's another thing that the foundation, led by Fred with Gail, has helped to kind of formulate. The FDA also helped with that, and we'll hopefully be publishing that soon. And that's really getting at this issue of, yes, different sites are using different clinical equipment, and how can we harmonize the parameters that they're using, the results that they're obtaining. That is a big challenge. When it comes to actual multi-center trials that would be FDA approved, you know, I don't know how they would feel if they would allow something that was using totally different clinical equipment. Obviously, most of the multi-center trials are run by the industry sponsors, in which case, you know, they're going to all be using the same device. If it was a large investigator-initiated trial, I don't know. I don't know if anyone on the call has any experience with that, if that's something they've thought about or they've discussed with the FDA before.
I guess that's exactly my question, in ways, the extent to which the parameters that were devised by the Focused Ultrasound Foundation with Gail and Fred, are they sufficient to allow different instrumentation to achieve those parameters? And so you have a harmonization at that point, whether it's a thermal degree or some MRI measured ablation level or something like that. Are we at a point where that can be used as a nucleus for multi-center trials?
It's a good question. It's definitely, I think, the goal of what we're trying to do with the harmonization. Fred, do you have a comment?
I mean, yeah, just to discuss these guidelines. I mean, I think that the difficulty that the team is pointing out is with ultrasound, and I think maybe this is a big difference between radiation therapy, for example, is that on one side we have the parameter, the device that we can calibrate, and we should calibrate. That we know that the device with such settings for treatment is generating such level of pressure, delivering such power, and blah, blah, blah. But then the bioeffects are really location dependent, tissue dependent, dependent on the depth of the treatment. And I think this is where it becomes more complicated. So if we think about thermal ablation, I think thermal ablation is thermal ablation. I mean, the difficulty is to assess the extent of the lesion. So if you don't have like the Sonalive clinical system, for example, an MRI guided system, it's difficult to really assess the extent of the treatment. So then I think you have to rely on surrogate markers, which will be your treatment protocol. I mean, I'm delivering, like you're doing, for example, for the breast, I'm ablating 30% of the tumor or 40% of the tumor. Or when you're doing histotripsy, I don't know exactly what the protocol is, but or you can have some feedback on cavitation levels. But I think that, I mean, and it's a personal view that we are still at an age where there are very few clinically approved devices. So that, I mean, the Theracleon, the Sonalive, for the prostate, you have two systems, I think. So I think it should still be manageable to have a multi-center trials and to try to harmonize at least the treatment parameters if we can't harmonize the clinical systems. But maybe as more Krit, maybe you have more experience than us.
Actually, if I may, I mean, Fred, you pointed out that the concept of dose will be elusive in focused ultrasound. I mean, at least in radiation literature or radiation dosimetry, we have some understanding of ionization as a means to look at, but in case of focused ultrasound, there are two aspects which I think the foundation may help us, all of us, especially people like me who are clinicians and immunologists trying to work through, need that guidance from the foundation. And so there are two elements which you discussed. One element is, let's look at the parameters of the device itself. And I think that can be standardized. That is very standardized. And yet, maybe the foundation, like you have for National Institute for Instrumentation, Standardization, etc., as well as in the radiation community, we have been working on centralized labs to do all dosimetry for every equipment and device so that, we can now compare each other. Okay, so maybe the foundation may think to help us standardize our devices, or at least whatever the output is and the parameters are, the set of parameters which we can now send back to the foundation to get kind of like a blessing that we are in the ballpark. But then comes the bioeffects, and that bioeffects is what we are all trying to define. And every lab has a different bioeffect. And the consequence of that bioeffect is the immune effect. So immune effect is so tertiary. First you have to have the bioeffect of what the beam did, and then so forth. So maybe you might consider having one RFA towards biophantoms. In other words, are there colorimetric or fluorometric activation? Like, for example, today the discussion of HSP promoter as an activator for thermal energy is one way. So maybe we can come up with strategies of biophantoms to define our bioeffects, whether it's a mitochondrial effect, whether it's a membrane effect, etc. And then you have already done the last one, which is the immune effects. To some degree, we have come that, okay, we would do some flow cytometry. Maybe we are doing some RNA sequencing. We are doing some TCR diversity. That part, we have lots of guidance from the community. But the first two elements, we have to guide ourselves. And I would be happy to offline brainstorm with you to see, whether the foundation can help us kind of get things together on this.
So I just want to clarify what you mean by biophantom. Would that be some sort of an experiment that's done that indicates that you get a certain bioeffect?
Yeah, so depending on what your endpoints are, and that can be coming from all of us, let's say, I'm just making up these, that we do have an unfolded protein response and we have a ER stress. So one of the effects could be a XBP1, which is a mediator of ER stress and protein unfolding response hitting a luciferase. So in other words, you can make a biological pathway activated fluorescent markers as a readout for the effects you are looking. And we can, I mean, I'm not just saying that it's one, but I'm just saying that at least that discussion, that can we really define the biological effects of focused ultrasound, because otherwise the concept of dose will be very hard. And Fred said it very nicely, because it is so much tissue dependent and so forth. But I would be happy to hear Dr. Moonen's, he has raised his arms, so let's see.
Well, this is just to come back to the question whether multi-vendor trials would be possible. There is one ongoing in our institution. That's not on immune stimulation, though, that's on ablation, but where we combine, it's a European consortium. It's the further trial that is led by my colleague Lenny Verkooyen, who's and there in that trial, there is both the Sonaleaf equipment and the Insight Tech equipment is being used. It's ablation, it's pain, it's about pain palliation on bone metastasis. So it is doable if you define it clearly what output you wish and how do you report it. And again, that's for ablation. It's a bit more simple because you can really image fairly well the effect that you have reached. So it's a bit more simple, but it is doable if you can define it well.
Thanks, Craig. Looks like Dr. Frank has a comment or question.
Sure. So can I comment on this a little bit? Because
Sure.
When we were doing large-scale MS trials on multi-institute trials, it was actually easier to get things done if they were all done on the same sort of MR scanner, the same sort of gadolinium contrast agents, especially if we were looking for subtle effects from a drug treatment on, say, contrast enhancement or even T2 changes or T2 star changes. You may find that same issue come up when we're dealing with the FDA, as them sort of wanting to minimize the amount of noise that could occur across vendors and want to stay with only one vendor. So if you're planning to do a multi-institute trial, you would actually see that, especially from an immunotherapy point of view. If some of our stuff is not sort of black and white, you know, it is not necrosis, for instance, with ablation. It's looking for subtle changes some places in the tumor size, or even on imaging tumor sizes or imaging edema, for instance, in various different treatments. So I can just point that out. It made it very difficult initially when we ran across multiple different vendors and we had to focus on one. So all the Siemens sites did all the one thing, and all the GE sites and all the Philips sites did one thing. And it became a lot easier, especially when it came to time for analysis, image analysis. And this may also play into, you know, looking at the bioeffects or looking at the effects of focused ultrasound plus X, Y, and Z treatment. And so that's just something to keep in mind if you're going to try to run a multi-institute trial.
Good points. Fred, looks like you have your hand up.
Yeah, just a last comment about guidelines, standardization. So as Crit, I think, pointed out, and again, I think for thermal ablation, it's more, let's say, quote unquote, easy to standardize. When we are moving away from thermal ablation, it becomes more complicated because, and I advertise the upcoming workshop we're organizing on cavitation dosimetry that will start at the end of September, where we are trying to convene the community to discuss cavitation dosimetry. And what we I think what will come out at the end of the workshop, which will be under several sessions to discuss instrumentation, discuss signal processing, discuss the question of dose. Is very complex to estimate because everyone has its own metrics to dose cavitation. the correlation between the different metrics and the bioeffects are not clearly understood. It really, of course, really depends if you do BBB opening or histotripsy or I don't know, pulse ultrasound. So this is in progress. And again, I think for thermal ablation, I would think that it will be more easy to standardize across institution, for example, for multicentric trials. When we are moving away from something that is more well understood, I think it will be more complicated. But we are working on this.
But at least even a consensus review by this group to alert. So, I mean, I was looking for our collaborator from FDA, but she is not here right at this moment, so I couldn't ask her this question. But my point is that, at least for us to understand that should we just remain in the realm of the device doing XYZ and leave it at that, or should we really look towards measuring the biological effects, because then that opens also a Pandora's box, because we do not know which one of those biological effects are actually going to be beneficial as a pharmacodynamic endpoint. And you were right, Fred, for heating, it's easy. We have done this for long time. And you can have MR-based dosimetry, thermometry, etc. But for these others, I think this is more of an investigation, and we should really collate the different parts. For example, some of the changes are really post-translational. So, for example, the Duke group have shown it, and we and others have also shown it, that STAT3 activation goes down. Now, that's a great point for a immune modulation, that if you give focused ultrasound, the phosphorylation event of STAT3 goes down almost immediately. So my point is that if we could make a list, even as a consensus review, of various measurable endpoints, which are the biofunctions, so to say, it can help anyone who is coming new to the field to realize that these are the specific changes one can look at and really try to correlate that with ultimate immune effects or not.
Okay. Natasha, did you have a comment?
Yeah, so just to start, I think Dr. Guha is making a really excellent point, especially for those who are new to the field, to have a set of comprehensive endpoints that people can look to, and perhaps that include things like imaging as well, which we were talking about before, would be very useful. And the addition to that too is again, we've talked about the variability that can exist in actually executing methods related to these endpoints. And that's something that I think the foundation can play a tremendous role in helping to shape and sort of standardize. But the reason that I raised my hand was actually to echo Dr. Frank's point, because I think he's making a very important point as relates to how, for instance, imaging is captured. And he laid out one rationale, but I want to offer one other, in that the foundation has talked about, bioinformatics, and I'll maybe specify imaging informatics in this case, and also database generation at some point. And as we start to think about these infrastructures that are going to enable us to ultimately perform big data related efforts, we are in a position of nascency right now, in the sense that we can position ourselves well to actually execute those analyses well. And what I mean by that is things like being able to discover imaging biomarkers down the road are going to be highly susceptible to parameters like scanner differences or differences in how contrast agents are administered. And of course, based even within a single institution, you'll come to find that there can be variability in these parameters, but a place where I think we have the ability to control them better is in the preclinical scenario. And again, I think that's an opportunity for the foundation to really offer guidance as specifically as possible as to how we can best control for these parameters, or at least to align institutions and approaches in a manner that allows us to collate data that lends well to AI approaches down the road.
Great points. Dr. Koklova?
Yeah, so I guess Dr. Gufa asked a really multifaceted question, but if you dissect it a little bit, and so if you think of, I think coming up with something sort of centralized as a database that this set of parameters broadly does this would be challenging for devices that are, let's say, one-off, that are put together in somebody's lab, but for platforms that are used broadly, or clinical devices that are used broadly, I agree that step one should be widely available calibration data, step one, with pressures all the way up to, and down the usable range that people use. Which, like a centralized database of just that, would be very helpful. Plus, and then further on, let's say you have a opinion system, for instance, which a lot of people have for preclinical data. It is possible, perhaps, to come up with sort of a if-then table that if you plug in intensity and depth of average tissue, and you could estimate, and the length of your pulse, perhaps, and you could estimate, I'm not saying measure and how precise that would be, but at least have a ballpark of what temperature elevation one may expect, because as Fred said, not everyone has an opportunity to measure temperature, especially in ultrasound guided approaches. So that's one, what acoustic radiation force and associated displacement could be. That's another one, which is also possible to do. And the cavitation is a weird one, because as Fred said, the dosing is all over the place, but at least the thresholds for nucleated cavitation or Hess-Tripsy type cavitation, or even boiling type cavitation, what those thresholds are, if known for those devices. I think that those would be helpful to have sort of in one place that is available to everyone.
Those are great, great suggestions. I think that this would, if we were to be able to do something like this, it would definitely require the community to get involved, as well as, I mean, obviously, I know the FDA has already been involved in a lot of standardization, as has AAPM and some other folks. So kind of not wanting to recreate the wheel and seeing what has been done, and then perhaps seeding grants or something for different sites to help with regard to different systems, because obviously who has access to all the systems? No one does, right? So but I see that this is a good thing to think about, so thank you for that. It looks like Petros, you have a comment.
Yeah, just about the standardization of the biological effects. In response to thermal ablation, it's easy to get lots of information in vitro. But with the histotripsy and histotripsy-like exposures, I'm not sure how easy it is to do these assays in vitro. So these are assays that can give us lots of information despite them being in vitro for the cellular bioeffects. And I don't know how easy it is to standardize them in vitro or even do them in vitro. I expect that the cells will just flow away from their substrate.
Dr. Ferrari, you have a question or comment?
Yeah, so this is kind of a different direction, but I guess if I were to look at a couple of pieces that seem like things that we should focus on in some way, one to me is for thermal ablation, because we can do thermal ablation, how to combine with therapeutics is something that we know is really powerful, and yet we haven't really pushed ahead and thinking about. So we know that we can increase the accumulation of antibodies or liposomes around an ablated site. And given that that we know the power of these immune-related antibodies, et cetera, and we can add in hyperthermia if we want to add in more. Anyway, I guess that piece still seems like it could use some kind of a focused thought, and particularly thinking about also whether whether there would be problems with denaturing antibodies if we were doing ablation. But it seems like it's such a powerful opportunity that it seems like one of the things that could use more attention. And I guess just listening to everything on the histotripsy side, seems to me large animal histotripsy seems like another thing that with really good immunohistochemistry could be also a really powerful addition to what's here.
Great. Dr. Allen.
Yeah, I apologize. I think I clapped last time instead of raising my hand. But so just in response to Petra's comment, we've been able to modify almost all of our studies for histotripsy for in vitro assessments. Sometimes the timing has to be adjusted. But one of the nice things about histotripsy is that we get a nice bubble cloud in a designated spot. And so you're actually testing the response of the surrounding cells that you don't actually treat. And so we've been able to conduct most immunological assays, everything we can do with other ablation modalities, we've been able to adapt for histotripsy.
Do we have any other comments right now? I think what we can do now is I'd like to ask Kelsey to share. She's been keeping a running list of throughout all of our sessions, the things that we've sort of identified as, next stage projects, things kind of on the wish list that we want to be able to do. And so now if we can take an opportunity to revisit these and then add to them as well. But even in revisiting these, if we can add if there's any more specificity we can add, things like, is this something that the foundation should be working on? Is this something that we ourselves should, are there folks out there who are really interested in this, that want to kind of participate in a collaborative effort, that sort of thing, as we're kind of helping to move towards building our roadmap for what we do next. And just to kind of say, out of this same type of discussion, in our last workshop came the ideas for the Immuno Guidelines, the treatment monitoring or guidelines, like we're talking about the kind of harmonization across sites between equipment. That all came out of these types of discussions, and the foundation takes all of this really seriously. So, this is your opportunity to sort of say, this is what we want you to do, or this is what we want you to fund, or we have this project that we want to do, and we think it's really critical for you guys to fund it. So please be open, and we'll keep track of all of this. Looks like Dr. Wang, you have a comment or question?
Yeah, thank you very much, Jessica. I thought it's a beautiful list. But I was also thinking, just by attending this workshop, I feel like we have a tremendous amount of opportunity ahead, particularly on the combinatory therapies. I like war games, so I thought, killing tumors just like, in the war game, people use like a bomber first, then tanks, and then soldiers, right? Perhaps to kill tumor completely, we probably can, leverage a similar kind of strategy, maybe using histotripsy or half first to kill the mass or the tumor core. And then we can, provide the second wave or controllable CAR-T by, guided by ultrasound, So I thought these combinations would be very powerful. And for us, we would love to collaborate with, expertise in histotripsy or half. So I would love to see whether foundation could help us to, foster these kind of collaborations.
Yeah, that's, I mean, first off, that's a really interesting thing. And I think we had that in some of our guiding questions. We never really got to it was if there is a role for kind of multimodality focused ultrasound treatments. So that's really an interesting concept. And then just as an aside, the foundation is always more than willing to help you establish collaborations with others in the community. So if, that's why we always encourage any of you in the field to talk to us as often as you like about not only the types of projects that you're thinking about doing that we may perhaps fund, but also if there are need for collaborators, we're more than happy to help you form those collaborations. And obviously, if we had this workshop in person, that would be one of the big things or one of the great things about these workshops in person. But we're happy to help you connect to anybody.
Thank you, Jessica. That's wonderful.
Okay, so it looks like Kelsey categorized these by sort of general comments or projects that people talked about, as well as in preclinical and clinical. So in the general, we talked about this precision medicine effort. If anyone has anything else to sort of add there, to add a little bit more specificity to that in terms of kind of next steps. Dr. Bullock?
Have you had any formative thoughts about how this bioinformatics core might work? Would you just sort of work with a particular institution that has the bandwidth to do that, or build off what Pisces been doing? And how would you be thinking that foundation funded science would have access to that?
So this was something that came up yesterday. So we have not been thinking about, or we haven't been talking about bioinformatics. I mean, I think from the standpoint of what the foundation is sort of capable of doing in terms of our core expertise and our bandwidth, I think it would require collaboration with some other entity or team that has that expertise. And that is something that we are more than willing to explore. But I definitely think it would require that sort of collaboration. I think from the Pisces standpoint, obviously, if we move to the stage, which we have spoken about with them in CRI, of if our preclinical work and our early clinical trials in, say, pancreatic cancer yield some interesting results, then perhaps focused ultrasound could be fed into their kind of Prince platform as a combination. And in which case, if it was, then of course it would kind of feed into that whole infrastructure that they have with the bioinformatics and everything. But aside from that, I don't know. We'd have to sort of explore if there's opportunity there. But if any of you have expertise in bioinformatics, or know of labs or folks that we should talk to, we would be more than happy to explore that. Or if just people have any more specificity on what they, with this idea for those who came up with this idea yesterday for the bioinformatics need, what would that need to look like? What kind of data, the size of data, or the types of data? I mean, this is not my expertise at all. So if anyone has any comments, Dr. Wang.
Yeah, thanks again. So I was thinking whether there's a possibility to, for example, share the name list of these participants or the workshop. That would be hugely helpful, right? People can start to look for possible collaborators in the future. At the same time, whether there is a possibility to create a shared document, for example, Google document, so that participant can list what they need, and then people who has expertise can sign up to discuss and form possible collaborations. For example, bioinformatics, there are expertise maybe here already, but we just don't know. So if there's such document, we can just all sign up and say what we were looking for and what we need, and people can probably answer in that same document.
Okay, so just a better better networking sort of application, whether it's a Google Doc, whether it's something else that allows people to indicate what their needs are, what collaborative opportunities they're looking for, that sort of thing.
Right, and people with
Yeah.
expertise, they can answer for these, needs, for example.
Mhm.
Okay. Yeah, so, I mean, I don't know if anyone had anything to add about any of the bioinformatics stuff, but if not, we can sort of just move on to some of these other things. Was to talk here about a forum to share clinical experience with FUS. I mean, I think some sort of a forum, definitely to have a more ongoing connection between the community would be good. So some of these are about developing connections. Obviously, we're happy to do that, as well as if any of you have connections with groups that you feel we should form collaborations with, please do let us know. Obviously, pharma is a still big kind of black box to us, so we need to do better there. And then this idea of this sort of biophantom development that Dr. Fuha was talking about. Okay, so I think we maybe should move on to the other ones just to make sure that we've captured everything, and then if folks have additional things that they want to add, we can fill those in. So in terms of clinical trials, obviously, there was this, as Dr. LaValle was stressing, looking at, with respect to with a focused ultrasound modality, what is a clinical hypothesis that we have there, and what data do we have to support that? So I think others had talked about if there's some way that the foundation can help, create some sort of not a master list, but like a good comprehensive landscape analysis of what is out there in terms of the data that we have supporting either different clinical hypotheses or other things as well. The window of opportunity trials. I don't know if anyone has any more specificity to add to that. I know that came up quite a bit. Phase 1 studies where there's a low barrier to entry. Longitudinal genomic studies. The idea of, specifically in a cancer like prostate cancer, where patients are already scheduled for a total prostatectomy, could we do something to treat those patients and assess the results before that? Anti-angiogenic therapy to normalize vasculature, to improve immune cell trafficking, and then add-on clinical studies to look at effects of FUS on the vasculature. I think additionally, I just want to stress that, this has come out of previous workshops, but this real opportunity that we have with focused ultrasound cancer trials that are taking place, the goal is not, immune modulation. And these are usually, a lot of these are ablation trials, or there are some that are the histotripsy trials now, but the opportunity there to add on immune assessment before and after focused ultrasound. And so that's something that the foundation is really, has really been supportive of. And so if you have any of these types of cancer trials that are in the works, please do come to us if you're interested in adding this immune assessment. And that's something that we can help to fund, because I think that any of the data that, the more data that we get in these sort of baseline situations, looking at focused ultrasound and its potential to inducing an immune response, that's going to be helpful as we move into trials where we're combining with immunotherapies. So I would just encourage people to also think about that. And then I guess looking at our preclinical, again, the identifying clinical targets and what's the hypothesis, and designing preclinical studies to validate if there anything, again, more specificity that anyone wants to add to that. This idea of designing preclinical studies to more closely mimic the clinical conditions or clinical trials that patients are having, so that we can get a better understanding of what focused ultrasound may be doing. Preclinical studies looking at partial versus total ablation, and then this anti-angiogenic therapy. I think it kind of in the second one, perhaps, is the idea of looking to some of the clinical trials, like, for instance, the Prince trial that Parker and Pisey have supported, and trying to mimic or design a preclinical trial that looks similar to that, and seeing what focused ultrasound could do in terms of improving results could have some utility. Dr. Bullock?
Yeah, Jessica, I think in terms of possibly the first bullet point there, there is an important opportunity to use some of the GEM models of induced or spontaneous tumors, but with the explicit intent of not trying to track a T-cell response at the end, because they're very challenging, because those models do not have a lot of antigens in them, but to really model the impact of the focused ultrasound in the very early stages, as Joe was alluding to a lot, the myeloid response and the innate response. The vasculature and the stromal setup of some of these GEM models is quite distinct from the traditional rapid turnaround transplantable models that we generally use for adaptive immune response assessment. But they are expensive and labor and time intensive studies, because it takes months for these models to come online. So that would be a powerful validation series of studies that the foundation could consider funding, to say, are we seeing different outcomes of histotripsy, thermal ablation, whatever, in GEM models that don't have a very accurate representation of the human disease.
Great. So now that we've sort of reached the end of this list, are there additional things that people think we haven't captured yet that would be good projects for moving the field forward? Again, this is your opportunity. Tell us what to fund.
I still think there's an important element of looking at lymphatic drainage as a function of the various degrees of ablation.
I would love to add the combinatory approach, or multimodality, combination approach into the list.
Definitely.
Thank you.
Anyone else?
I'm not sure how this overlaps, but I think that if we were to try to be administering therapeutics before ablation to understand how that would impact both accumulation, clearance through the lymphatics, and efficacy, would be
yeah. Dr. Kuklova?
Yeah, I guess it would be tumor specific, but in terms of combinatorial approaches, I think it's worth thinking of when devising the combinatorial approaches, it's worth taking into account what the standard of care therapy is right now and sort of mimicking that. So for instance, just combining, say, checkpoint inhibitors and FUS is not going to be realistically representing somebody who has been treated with chemo for a while. And so just basically including all the other, or as many of the other therapies as possible into the mix that is currently being used sort of for as advanced disease, I think would be important.
Thank you. Fred?
Yeah, just maybe a question for the community. We discussed it quickly, I think, yesterday also, about the comparison of modalities and the consortium project that the foundation has funded in the past on the GBM. So do you think there is value in funding setting up other type of consortium studies where we select a model and we compare the different modalities? And if you think so, what do you think would be good targets, good models?
Fred, before anybody comments, I had a suggestion on this. So, there are situations where we need anatomic location, like GBM for me is an anatomic location for now, and then of course the biology. Bone mets, same thing, the bone met palliation versus bone met treatment are very different. So there are some anatomic challenges for ultrasound, and maybe one of the RFA could be related with anatomic challenges. If we really think ultrasound will play a role in immunotherapy, especially in metastatic diseases, there are two indications, either soft tissue or bone. And so maybe you can split these requests for funding. Let's say, how would someone create an ultrasound device or an ultrasound treatment parameter to treat bony mets? Okay, which is a challenge. And but on the other hand, if we have to play a role, we will get these type of metastatic settings, which are normally not prepared for ultrasound, and it's a challenge. So that's one which is related with anatomic question. But the second thing is that we could at least split the question in hand into what Kathy says so well about priming versus ablation. These are two separate concepts. So we could sometimes request priming grants, which doesn't necessarily go for ablation, but prepares the tissue towards an immune effect. So it's more of a priming grant versus ablation. And these are two separate applications which will have totally different ways to measure their effects. Okay, so it just doesn't have to be always anatomic, like by saying that we want to tackle pancreas or GBM, but it could be anatomic with respect to our challenges with ultrasound. The biggest question in my mind is ablation good, or is ablation enough, or priming necessary? And I obviously belong to the priming category, that you can ablate, but prime first before you ablate. So we could, based on our own experiences between all the different groups, see if we were trying to do priming, what are the parameters we are thinking of priming as an example? Is it antigen presentation? Is it immunogenicity? Is it antigen processing machinery? We can attempt in breaking down that priming into certain columns and now have different labs participate, that maybe I'm only going for epigenetic modulation of antigen processing machinery. someone else is going for, like Kathy had done with CpG oligos, activation of dendritic cells or reprogramming of myeloid cells. So we can divide this up and then say it's a consortium grant where immune priming will be analyzed this year. And these are the things we are going to tackle. Versus if we are going for ablation, then the immediate impact is thermal mechanical. We have all discussed, and Kathy so eloquently put the summary in the email yesterday. And that's a good read for anybody who is trying to follow the story of ablation. And again, you can split it up in terms of immunological domains for a proposal and tackle it. Because as far as the tumor microenvironment is concerned, that is more or less a bigger question where everyone else is also participating. But as far as the device is concerned, we are really thinking of these two features, priming versus ablation. That could be one way of looking at it.
Thank you. We just have a few more minutes, so any final comments or thoughts before we close out? All right. All right, well, we can give you a few minutes back. So I want to thank you all so much for joining us for this 2 days and really engaging in the discussion. I think this was the best of our 4 workshops that we've had in this area. And it's really thanks to all of you for actively engaging in our discussion. It's clear that the field is maturing a lot, definitely in the last couple of years, but we still have a lot of open questions and we have a lot more work to do. And the foundation is, of course, always here to support you all in collaboration with our partners. So again, I want to thank you. I also want to mention that if you had to miss any of the sessions, they will be available on the workshop platform by the end of the week. And so that will include all of these live sessions as well as the presentations that were played live. So again, thank you all, and stay safe out there, and we will be in touch. We'll of course we always do a summary for these things, a white paper. So we'll be in touch when we have a draft of that to get your comments on that as well. Thank you so much.
A huge applause.
Right.
Thank you.
Thank you.
Wonderful.
Thank you. It was good
Thank
seeing
you.
everybody.
Bye-bye.
Thank you.
All right, bye.